So my name is Vijay Kuchroo. I'm from Brigham and Women's Hospital, and basically do basic research. And I think one of the questions that we have been dealing with, how do we use computational tools and technologies to better understand the biological problems? In fact, with whole genome on our fingertips, we are in a position to identify not what happens to one target with one drug, but with the whole genome. So that will be on one aspect where you can use the computational tools and technologies to understand both the effects and side effects of your drugs, on-target and off-target effects. But you can also use the computational tools and technologies to, in fact, identify the target and functions of the target. And I think we're going to talk a little bit more about it during our discussion.
Yeah, you bring up a really important aspect of integrating knowledge in this genomics era, as we're able to generate not just a lot of bulk genomics, proteomics data, but also on individual cells and on their spatial location, which allow you to use computational biology and increasingly also generative AI to interpretate and to see patterns between diseases. I'm really intrigued, and we were actually talking as we walked up here, that some of the pathways that are active in different ways the immune system fight off threats, whether it's an invading microorganism, whether it's altered cells like a cancer cell, or whether it's misguided autoimmunity, some of the pathways may play a role by either being suppressive for one outcome, or you need to have it agonized, activated for another outcome. And we were just chatting that some of the more profound cancer pathways in immune oncology, CTLA4, PD-1, indeed do play a role also in how the immune system acts for inflammation, autoimmunity. So I think we would gain a lot by deploying the new tools of data sciences, AI, to identify which pathways do seem to play a very important role in various types of immune response, which tissues, which outcomes, and we're missing out by still working in silos. So this is a big opportunity for the future. We're investing quite a lot in trying to understand key pathways, the zip codes, which of the molecules, ligands, receptors, in which tissues, and what represent genetical susceptibility versus what represent acquired changes at the epigenetic or protein levels. And finally, of course, AI for therapeutic outcome, it's not just understanding where to intervene and how to intervene, but also how you design your molecules, where we have had a tremendous use of AI from a number of projects. Most recently, our poster child project, Paxlovid, for intervention in COVID, where AI played an instrumental role in designing molecules with high exposure in blood.
Yeah, I mean, just actually, I'm going to follow up on what Mikael said. Just imagine for a minute that we have been spending so much time first identifying the target and then crystallizing it, which takes multiple different years, then identifying the pockets in which a drug could fit and interfere with the target. And I can imagine in the future that with all the computational tools and technology that are coming through, that AlphaFold is just one example of it, that if you have a linear sequence of a target, you can put it through the AlphaFold. It will tell you exactly how the structure of this molecule is. It will tell you what the pockets are, where you can fit different kinds of drugs. And then we also have the whole library of the drugs and the molecules and what their structures are. And you could actually imagine how do these molecules actually fit inside these pockets, and will they interfere with the functioning of this pocket? This is even without lifting a finger. You don't have to do any experiments. what was taking 5 to 10 years could be actually shortened into months and maximum a year, that you have a linear sequence. We know it's an important target. There's a mutation in the target. You can put it through the AlphaFold. AlphaFold will tell you, in fact, what the structure of the molecule is, what are different pockets, the enzymatic pocket, and this is the relevant pocket that has been inhibited. You have a structural library, and in fact, a computational program will tell you what are the best fits for these pockets, and you can interfere with the functioning of the drug without actually having to physically do anything. All you got to do is to take this data set and the drug and the target and validate whether this is true or not true, rather than actually doing everything from scratch. And this is the power of the computational tools that are coming through the pike. And not only that, once you have made a drug, that if you do a simple genomics analysis, you can immediately figure out, is your drug hitting the target? If it's hitting the target, is it actually blocking the right pathway that's relevant for the disease? And is it affecting anything else outside this target which you may be giving you side effects? And you will know immediately the fingerprint of the target and the drug and the side effects and on-target effects.
That's a great, both you guys, that's a great characterization of the use of AI. And I think if you think about the other applications, Mikael, you mentioned Paxlovid, but let's talk a little bit about diseases of aging. And this is obviously in the U.S., and throughout the world, this is going to become a much larger population to address therapeutically. Talk about the challenges that you're looking to at Pfizer to address diseases of aging, just with respect to either neurodegeneration or even immunology.
Thank you, Geoff. We really got intrigued by the challenge during the pandemic, where you treat billions, to look at some of these larger populations. And it's clear that the aging population is growing rapidly. By 2050, we expect the population above 65 to be twofold bigger, the population above 80 threefold, and the population above 90 fourfold. Aging is one of the major risk factors currently for a number of diseases. There are several cancers where we certainly have been active, like the most prominent in men, prostate, and in women, breast cancer, that increased by aging. And it's likely a combination of where Vijay was going, how the immune system changed by age, and it may go from being a more eager to learn and active, vigilant immune system to a more senescent, less eager to be triggered immune system by age, at the same time as cancer cells, whether breast, prostate, and even so more in lung cancer, accumulate more and more mutations. So that's why we see a dramatic increase in cancer because of the aging population, although we, as a life science community, have introduced many good drugs. If you look at cardiovascular diseases, aging is another prominent risk factor, probably related again to immune dysfunction, linked to, of course, environmental factors, whether lifestyle, diabetes, lipids, combined with immune dysfunction and chronic inflammation in vascular and other metabolic tissues. And of course, finally, going to the big challenge with neurodegenerative diseases, where we've learned that a major hallmark is not just deposition of amyloid and tau. Even if we remove those by monoclonal antibodies, the benefit is rather moderate. It's really the response to those that have progressed into a chronic, difficult, scarring type of response that one needs to intervene with and characterize the main immune actors, the pathways. So being able to create that encyclopedia in a digital fashion and not just understand the written chapters, but use generative AI to predict how it evolves with age, it's going to be such a crucial opportunity for us.
I mean, I could just add what Michael just said. Obviously, we're all going to age. We are aging. And the population of that's going to be over 65, obviously, is going to be increasing at a dramatic rate. And in fact, in the Western world, we don't have younger populations. Actually, we are aging, especially both Western Europe and the United States. And the immune system is probably at the heart of it, as Michael said. When you're a young adult, your immune system is very active. And that's when you see most immune-mediated diseases. All autoimmune diseases come in young adults. But as the immune system ages and it becomes partly dysfunctional, and that's why in COVID, you had to give multiple vaccines to elderly, and they died at a faster rate as well. It's because the immune system is not responding at the same rate as it does in the young adults. And that's why you see neurodegenerative diseases much higher in aging population. Cancers, because they can't be detected and eliminated as your immune system actually becomes older and dysfunctional. So we think the immune system is at the center of this all, which actually people don't really think that way right now. And you have these stem-like resource cells that are there when you are younger. And as you're old, you lose those cells with time. And the cells that you have senescence or dysfunctional phenotype. It's almost, these immune cells look like as if they have gone through, immune cells for an elderly age look like they have gone through multiple different cancers. In fact, you can see the phenotype genomically, even if they have not experienced anything, they look like they have decayed over time. So how do we actually develop healthy aging? We are all aging, but nobody is thinking about how do we do healthy aging? That the lifespan has increased, but is it healthy lifespan? We're all saying that we have increased our lifespan from being 65 to 75, but are those years that we have, are those healthy years? And how do we actually get to that stage? And how do we actually keep our resource cells alive that replenish these effector immune cells that will fight against cancer, that will fight against infections like COVID? They will provide an immune response to vaccines, and they can also, in fact, help us detect like the amyloid plaques in the neurodegenerative diseases. In fact, we think the immune system may be at the heart of all these processes. The other aspect we haven't actually talked about, which is really even more interesting, is that the immune-mediated diseases are on a rise. And we don't, in fact, actually even talk about this. That if you really look, is that every other kid in a classroom has allergies. They are carrying EpiPens in their pockets. In my generation, nobody carried EpiPens. In fact, at a single peanut, there's these anaphylactic shocks, and they land up in the hospitals, and kids die because of anaphylactic shocks. And why is it that in these 2 decades or 3 decades, our genetics has not changed? Why are these immune-mediated diseases on a rise? And every other kid is carrying EpiPen in the pocket. In fact, I have heard mothers telling their kid, Billy, they don't ask them, did you carry your food with you? Did you carry your lunch with you? They say, do you have your EpiPen? And I have heard it myself. So it is a problem, but what is the source of the problem? It is not the genetic change that has happened. It is something in the environment. You can see that IBD has gone up 5-fold in 3 decades. Multiple sclerosis has gone up 3-fold in the last 3 decades. And asthma allergies are just on a rise. And this is what Mikael Dolsten was talking about, these immune-mediated diseases, and they come in young adults as a prime of your age. And how do we detect and control this rise of epidemic? So at the young adults, you have the issues of immune-mediated diseases. as the population is aging, and the aging is not healthy aging. In fact, we are dealing with increasing number of cancers, neurodegenerative diseases, and lack of responses to vaccinations. And we have to do something about it.
Just a quick add-on. You spoke about healthy aging. It's interesting in this era where vaccine has been so instrumental for us over the last 3 years that we learned, as Vijay alluded to, if you vaccinate earlier in life, you get a very vigorous and often a broad clonal response. As you age, the magnitude of the response is usually less, and the number of unique clones, T and B cells, are more limited. So it's an investment for healthy aging by being able to train the immune system very early to recognize a number of pathogens and then continue later in life to evolve and more sophisticated. And we certainly hope that paradigm can be broader discussed in society, because today it's often that we wait to vaccinate until the risk to get the disease has increased. Like recently, we and others are introducing RSV vaccines. We introduced pneumococcal vaccines for the older population. But actually, the best would be to experience those vaccines earlier in life and be even better prepared for healthy aging later on.
Right. Yeah, let's explore that a little bit more, Mikael. So when Pfizer's leadership in infectious diseases really predates even COVID, right? With some of the vaccines for pneumococcal, but maybe talk about how at Pfizer you're able to further advance your platform in infectious disease as it relates to again to this theme of diseases of aging.
Well, I think as we need to vaccinate against more and more pathogens, for example, we recently got our 20-valent pneumococcal vaccine approved both for infant and for adults. Here we have national immunization programs for the youngest, where you start early in life and continue until you are 1 year, and then you can boost when you're at the later stage. That's an example where you get maximal effect of a vaccine. And as we add more and more serotype to protect to more and more pneumococcal strains, for the older populations, we need to refine more and more how we present those vaccines. New technologies, what we speak about, new carriers, click chemistry, and likely for some very complex vaccines, adjuvants. We try to avoid, if we can, adjuvants, which we did very well with our RSV vaccine. Vijay alluded to structural-based understanding of immune reactions. That was deployed for our RSV vaccine to optimize the immunogen for the immune system. Sometimes it's hard to do that because there's so many immunogen, and then you can add for the older population adjuvants. One example is completely new technology that directs multiple arms of the immune system. We all take a vaccine for flu, and unfortunately, it's a little bit like a hit and miss. Maybe every second year we get well protected, another year we get just somewhat an attenuation of the disease. It's a very old technology that relates to growing virus in eggs, or possibly preparing over many, many months an immunogen in cells. Now, we are deploying right now the learnings about mRNA vaccines to very fast be able to design the most relevant for each season's hemagglutinin and allow it to deploy it so both B and T cells reacts, and allow you to mobilize more than one defense line in order to manage a flu in an aging population. So it's really interesting to see, as we discuss here, better insights into the molecular regulation of disease, better technology to provide new vaccines or medicines, and the big data sets together with AI that allow us to bring together all the data points for what each age group may need.
One of the, just following up on what Mikael said, obviously immunology and understanding immunology in the last 20 years has actually grown dramatically. And we know that you have the innate immune system where the adjuvants play a very important role, and then the adaptive immune system that gives you memory. Once you're infected, you are immune because you develop this memory. Like in brain, you get the immune system also has a memory. And whenever the infection comes back, the cells from the memory will come back. One of the things that has not kept the pace is the technologies to develop new adjuvants so that you can evoke the full potential of both the innate and the adaptive immune system. And that's what Mikael has been talking about, is that I don't think the drug companies or the vaccine companies have actually fully enlisted both the innate and adaptive immune system in such a way that you get immediate protection and then you get effective memory. It's only now have we actually begun to really understand what is it that is safe. The adjuvant is safe for the human being. It is activating innate immune system and is giving effective memory later. And I think as the technologies are coming forth, as we are learning more about all the components of the immune system, and also better ways to make vaccines. I mean, mRNA vaccine was a revolution, that you don't have to actually make a full protein and combine with an adjuvant to give a vaccine. And normally a vaccine would take 5 to 6 years to develop. This was a remarkable feat of science, that you could take an mRNA and use it in an effective way to evoke an immune response. And that's actually where the science had really proven to be so useful. And thank you for that. In fact, Pfizer was a leader in this process, and really grateful for all the work that you guys have done.
In the last few minutes here, we've talked about infectious diseases, but Mikael, I wanted to think about the other applications of what you're talking about, next-gen technologies, not in infectious disease or immunology, but more like, let's say, in metabolic diseases, diabetes, obesity, et cetera. Talk a little bit about Pfizer's sort of emphasis on that.
Yeah, it's clearly another non-communicative disease pandemic, diabetes and obesity. It started in a Western society, but it's even globally migrating to be a major issue. It's not just how much we eat, but what we eat. And it's a really integrated problem, because food nutrients seems to interact a lot, of course, with our own metabolic systems, liver, fatty liver, adipose tissues that grow in size and accumulate inflammatory component, the vasculature that becomes inflamed. But we also have a chronic phase where the microbiome community of bacteria becomes less healthy. So you have the genes of the bacteria, you have the epigenetic changes that are induced in our own cells that then lead to a more chronic, difficult to treat metabolic dysfunction. One of the areas that have been so impressive in recent years is how we are able in some way to reset the set point for satiety and for weight by moving the needle for some of the endogenous signals. And the GLP-1 that was originally pioneered by a company using peptides, it's an original peptide that's released in the body, and many companies did once a day and once a week. Now, for many metabolic diseases, I think there is a hurdle to go into injectable, and you want to have an opportunity more to have a convenient lifestyle. So we have been able to use some of these sophisticated technologies to design oral drugs that can do the same as an injectable peptide. And from us and other companies, we're seeing that these oral GLP-1s can bring down, if you have high HbA1c as a diabetic marker, dramatically, more than any other oral medication, and can bring down body weight dramatically. So we see a future where completely new insights can be used to treat in a convenient way and maybe help us to intervene earlier in life than feeling that we need to move on to an injectable treatment, which often is having a psychosocial affection of that you're having a chronic, irreversible, life-threatening disease. And we are very eager to move more towards primary care, that type of medicines like an oral GLP-1, in the same way we are documenting other drugs to show that you can treat and reverse diabetes, you can reduce body weight dramatically, prevent fatty liver, save the kidneys. Now, we are used to go to an annual health check and look at blood pressure, glucose. We may in the future be looking at the levels of these satiety hormones, but what would be intriguing is to also look at the immune age as we come to the doctor. There can be a big difference in immune age. It's not what is said in your birth certificate when you're 50 years old. You may be 35 years old in your immune age, or you may be 65. And like Vijay spoke, if you have a very young immune system, you are more predisposed, likely to have a late onset of an autoimmune disease. If you have a fading immune system, you are to be monitored for vascular disease, neurodegenerative disease, cancers and infectious disease. So the future should be really about understanding insight, not looking at historical things like your birth certificate 50 years ago.
That's a good way to end it. So thank you very much, guys.